And so in the last 5 to 10 minutes here, I mentioned that we would talk about human herpes viruses. And so this is a very different kind of virus than anything we've talked about so far. And despite that, you can see in these little schematics, they look kind of similar. You've got an envelope protein, you have an envelope, it's still enveloped, you've got a viral capsid, and you've got the viral genome inside that viral capsid. The viral genome here is one of the things that's really, really different in that it's a DNA genome. This is the only DNA virus we're going to talk about. It's also gigantic. These viruses are much, much larger than anything we've talked about by at least an order of 5 bigger than coronaviruses, but an order of 10 bigger than flu viruses. So these are much, much more clever viruses. They have a lot of tools. They have, in many cases, over 100 gene products. So we go from 11 in flu to 120, 130, 180 in some of the herpes viruses. So they're gonna mess with our immune systems much, much more. And as a result, they're gonna be much more successful pathogens. These are the 8 human herpes viruses, HSV1 and 2, which we often call just herpes, and that's associated with skin-based sores. Varicella zoster virus, which is VZV or chickenpox virus, HHV3, related actually to HSV1 and 2. And if you had chickenpox, you can spot that relationship. EBV, which is mono, HHV4, it's a gamma herpes virus, and somebody here is working on gamma herpes viruses, which also Kaposi sarcoma, which is HHV8, also a gamma herpes virus. EBV, lots of people have. KHSV, not that many people have, or less people have, but it's also not looked at that much. It's easier controlled by your immune system, and where it really becomes a problem is in people that are immune compromised. And so it was one of the things that alerted epidemiologists to the rise of HIV, because people with KHSV started developing tumors after their CD4s were depleted. And then HHV6 and 7, which are super common and cause like rosacea in infants, almost never studied outside of that, except in cases of immunocompromised. Hugely successful viruses. You hardly anybody studies them. I think it's a super interesting understudied area. And then probably the best studied herpes virus, which is cytomegalovirus. Estimates range that about half the world is infected with CMV. It's definitely more than that in certain populations, and it definitely accumulates with age. And we've done a lot of work on looking at the prevalence of the different herpes viruses across populations, and it's actually quite surprising. They vary very widely, and they don't correlate with each other. So some populations are very high in CMV, some EBV, some both. Cytomegalovirus causes something like a mono-like disease, but otherwise, EBV, CMV, you can carry for most of your life without being aware of them past that acute phase. So all of these can be chronic infections, and their mechanisms of chronicity, I would say, are something closer to, for everything but the alpha viruses, are something closer to HCV, where the virus is hiding out in the nucleus, so not like HCV, which is in the cytoplasm, but goes into this circulation of its genome, and it just keeps its genome in the nucleus. If the cell divides, the herpes virus DNA divides with it, has a very complex replication cycle because it's a gigantic genome, and so it needs a lot of resources to replicate its genome versus something like flu or even HIV. And then we'll make viruses and release them in the same way. So enveloped virus needs to make its proteins assemble on the cell surface, bud out from the Golgi, and so forth. The alpha herpes viruses have what's a real latent period where they will shut down most of their transcription and have a very special form of transcription that can then reactivate and turn into rapid transcription, translation, and production of new viruses. During the latent period, they're not making new viruses. And this is why, if you think the alpha viruses are herpes and VZV, that's where you get these sores, because when they become really activated, they're lytic and they're killing the cells that they're in, and they're causing a lot of painful inflammation. In contrast, something like CMV, which is let's say our most successful herpes virus, doesn't do this lytic latent switch. And it's described in all of the textbooks as a smoldering infection. And so that just means, again, it's kind of like that HCV idea where it's just kicking itself over time, not super high levels, but not super low levels, and just sort of ever present. And so that's just kind of summarized here, showing the HSV lytic latent switch versus CMV, which just kind of hangs out. If the cell gets stressed, you might make more CMV. If you become immunosuppressed, you make a lot more CMV. And most of us, if you have CMV, it's very hard to detect. You can look in the saliva, you can look in the urine, very, very hard to detect. If someone gets a bone marrow transplant or has very late stage HIV, then you can detect genomes. But before that, it's this smoldering low-level antigen production. And EBV is a little bit similar, but EBV hides out in B cells, and if the B cells get activated, then they'll upregulate significantly. So why are these interesting? It's because they have to undergo constant immune surveillance. And so anything that compromises your immune response will allow herpes viruses to become a real medical problem for people. So they can be fatal in situations of profound immunodeficiency, in organ transplant, late stage HIV, bone marrow transplant. CMV, EBV are often causes of death in these individuals. And even in healthy individuals, we can see the evidence of the pressure that CMV is putting on our immune system. This smoldering nature of CMV provides a persistent source of antigen, but it doesn't drive exhaustion of the immune response. It drives this sort of inflationary expansion of the immune response. And so people that are CMV infected, a subset of them, not everyone, and this is a big area of research right now, have this inflationary T cell response, which means, in your early days, you make a memory response, you're controlling the infection. There's lots of chronic infections where you kind of have this low level of T cells, even HSV, toxoplasma, these infections where the antigen, even TB, the antigens are always present, but you make enough T cells that keep them under a reasonable amount of control over decades. In CMV, what you see in a subset of people is this slow climb of the number of T cells, particularly CD8 T cells that are specific for CMV antigens, to the point that in humans over 65 years old with CMV, the average portion of all of your T cells that are CMV specific is at least 10%. Sometimes it's 20, 30, 40, 50, up to 80% in some individuals of all of your T cells become focused on CMV. And this is for a disease, for a virus that most of us don't even know we have, but your immune system is really focused on this highly distributed virus throughout your system that's making very small amounts of protein and very small amounts of new virus over the course of your life. And just concluding, all of this is, again, still this type 1 immunity. And so we need interferon gamma, IL-12. We've got a CD8 T cell that's going to be surveilling the endothelial cell or the target cell that the CMV is infected in. It's going to be instructed by a CD4 T cell. So that's why that whole Th1 CD4 apparatus has to stay in place for us to control these infections and why they become problems with HIV. But also, if you think about this network, if 80% of it becomes given over to dealing with just CMV, it's going to be its own compromise of other responses. And so this is one of the theories in elderly individuals for why they do a very bad job of mounting new responses to things like SARS-CoV-2, because their immune systems have now become so distracted with these other chronic infections that they've devoted all of their resources to. And so that's the last slide I have for this section, and I'm happy to take any questions.